All right, now, as I stated, there are these databases offer different types of graphs. Yeah, if you want to ask a question out of the scope of the course, feel free to write in the chat box. I'll be happy to answer. Oh, what is arrays, Burçak? The chat box. Oh, what you were writing? Okay, okay. If you feel free to write it again. Okay. Okay. All right. So these are different types of graphs that we see here, uh, uh, like. In pathway commons, it's apparent we have, for example, physical protein interactions, gene regulatory interactions. We have, for example, controls phosphorylation of different types of physical interactions, which some of them, like incomplex with, maybe stationary interaction. This one may be like a transient interaction in which A1, BG, phosphorylase AKT1, and that's it. After that, may be a transient interaction that happens in a very short amount of time. And we have also interaction between edges between, for example, two genes that say CA2. It catalysis precedes this one, so they catalyze a chain of reactions. And here, whatever this gene is, CA2, the reaction that it catalyzes, it acts as an enzyme, comes just before the other reaction that is catalyzed by LSG1. So this may also, there's no direct interaction, physical. This is not an example of a physical interaction. They do not interact physically, but in a biological pathway, they come one after another. You may also want to visualize, if you want to visualize a metabolic network, you may want to put an edge between those two. All right, so as you can see, there are different ways. And what I'm going to show you next is how can we represent this graph data to do something about them. For example, if I ask you, this is just an edge list, by the way. Many of those things that we have downloaded the BioGrid data set. Let's also try to extract it here. They all come with, as you can see, a zipped format. And I say extract here, wow, it's even going to be larger. So let's see. This is the BioGrid data. Let's look at its properties. It's 1 gigabyte almost. Okay, so this is a 1 gigabyte interaction file, but we'll see. Probably Notepad++ is going to struggle a lot, but it will be able to open it. Another thing to do this, I mean, obviously you're not limited to Windows. On Ubuntu, for example, for these large files, which are gigabytes of files, but as you can see, Notepad++ is able to open this 1 gigabyte text file easily. I think if you try to open it in Excel, for example, it will struggle a lot, or some other tools. But here, it's a huge file in the sense that this is a single line. A single line. There's the BioGrid interaction ID, interest gene ID for interactor 1. So this interest gene ID protein gene interacts with this interest gene ID interactor B. This is the BioGrid interactor ID. BioGrid also gives them separate IDs. This is the systematic name. They don't have any systematic name, but this is the official symbol, official gene symbol. So if you just are interested in who interacts with who and the gene names, these are the two columns that you're interested in. And this is a tab separated file, as also suggested by the name. There are also other synonyms for interactor A, synonyms for interactor B. I told you that the same gene can be referred to with other gene symbol names. For example, MAP2K4 is also known as, if I'm not mistaken, JNKK, or in some other article, JNKK1, or MAPKK4, or MEK4. Okay, so look at this. These are all synonyms, also known as type of names for this one. Also, FLNC is this. These are the synonyms. So another thing, what is the experimental system that identified the interaction between these two genes, two hybrids? There's as I told you, biological experiments performed specifically to identify whether two proteins interact or not. One of them is the yeast to hybrid mechanism, in which two proteins are there's a bait protein, a target protein, and there's a known reporter protein in which certain fluorescent signal or some reporter signal is going to be observed as a phenotype if two proteins interact with each other. And they just tie the proteins that you're looking for to these reported proteins, and they and if they do physically interact, the two hybrid system shows you that there's an interaction. So this is the type of interaction, physical interaction, experimental system type. Author, publication, source. There's also an article with a PubMed ID that tells you where this evidence is coming from. As you can see, the BioGrid information contains lots of additional information about these interactions. That's why it's 1 gigabyte of file. But in terms of the number of interactions it contains, it's around 2 million. So 2 million physical interactions. Now, you may ask, why when this is having 2 million interactions, why pathway comments does only have 1.3 million, since it claims that it gets some data from BioGrid as well? Maybe there may be some redundant. Two proteins may appear in more than one line in BioGrid. I don't know. I don't know the reason of this actually. I'm just asking this as a discussion point. There may be several reasons. One of them may be, as I said, the same pair of genes may be present in this BioGrid list more than once. If it appeared, for example, in two separate articles, they will probably have two rows, two lines for that same pair. That may be one of the reasons. But as you can see, all these three different protein interaction database downloads, they all have one thing in common. They are nothing but edge lists. What's a graph? A graph is mathematically, a graph is a tuple, which is a set of vertices and set of edges. So V indicates the set of vertices here, and edge is all the list of edges is also a set of tuples. For example, vertex 1, vertex 2, which indicates there's a, so it's a list like this. So each element that you see here, it corresponds to a line, a single line of your graph, of your biological graph. So if edge list is only sufficient to represent this graph, why do we have the vertex list V here to represent mathematically what a graph is? So what I mean is that all our data sets are provided actually in this format, where every line corresponds to an item, an element of this set. So in other words, if you're going to use this, for example, string graph, where there are physical interactions between proteins, you will have a set, an edge set, whose size is 8.6 million. So there will be 8.6 million elements in this set. But what about the vertex set? How do you find it? By looking at this file. Or another thing is that, why are we just given the edge set? Why are we not given the vertex set? Or if edge set is sufficient to represent a graph, why do we have mathematically, why do we have this vertex set also represented separately? in graphs. Any comments on that? If you look at the edge set, the vertices appear in the edge set as tuples, right? So the edge set also contains, if I just go over this edge set and maintain a list of vertices as I see them. Oh, there's V1, there's V2, oh, there's V3 here. So I can actually maintain a vertex set and construct a vertex set while I'm going over this edge set. Usually these files are processed like this. You process it line by line. Even these 1.3 million interactions, while you're processing it, you can maintain a map, a hash map, hash table of vertices, which will its size at the end. Every time you see a pair of vertices, you insert them to your hash map if it doesn't already exist in your hash map or hash table, whatever you're using in what language. But in the end, you will be able to construct a vertex like this. The reason in real graphs we have this second vertex set is that there's some disadvantage of this edge, not disadvantage, but a shortcoming. There's a shortcoming of such an edge list. It will not be able to have certain vertices if they exist in the graph. For example, what if there's a gene which is not connected to anybody in a graph? Look at this. I have a gene here, but it has no connections in this biological network. Is it possible to have this gene in your graph in an edge set? No. Okay? Singleton vertices, which means that vertices which do not appear in any edges, or vertices with 0 degrees, with no edges, this kind of an edgeless representation are unable to represent them. You may say that maybe we can have an edge to itself as a self edge like this. I can have it appear in this edge list as GG. But actually this have a meaning, a biological meaning. If the gene interacting with itself doesn't mean that it doesn't have any edges. It means that it forms a homodimer with itself. So what if I have a gene which doesn't have forming a homodimer? So I really want to have a gene with no interactions whatsoever. This edge list will not be able to have those genes. So that's one thing. In real graphs, for example, in mathematical graphs, we may have vertices that are not connected to anybody else in the graph. So in order to represent those, we need to have a separate vertex list in order to have such graphs. But in terms of biological networks, I think everybody decided that if there's a gene that doesn't interact with anybody, it's okay that it doesn't appear in this edge list. I'm not going to put that gene into my analysis anyway, because that gene is not interacting with any other gene. And maybe such a gene is such a gene possible biologically, a gene that just functions on itself, not interacting with anybody else? Probably not. I mean, just imagine an electrical circuit board in which there's an AND gate which is not connected to anywhere else. What is the purpose of having that AND gate or that gate in your circuitry board? Okay, if you put something in that board, it has to be connected to at least one of the elements of the circuit, right? I don't know, maybe there may be some function of it to occupy space or maybe to do something else. There may be. So, but I'm just speculating on why biologists decided to have just this edge list and not a separate vertex list. Some programs that visualize graphs actually take node list and edge list separately. Okay, so not every graph input format is just an edge list like this, but we have looked at 3 biological network examples from BioGrid, from String, from Pathway Commons, and in all 3 of those we have seen that the interaction is provided to us. The biological graph is provided to us as an edge list. All right, so what is the best way to read this graph into memory, or whether is it going to fit in memory? Let's ask this thing. I mean, if you look at the sizes, the BioGrid one, which is 1 GB. I mean, the whole text content should be able to, we should be able to fit it in memory. It's 1 GB. So we usually have like 8 GB of memory available to us most of the time. But let's start with the simple one, the smallest network, but it's still, you see, it's a large network. First of all, we need to determine what kind of a data structure are we going to have to represent this information. Are you going to, if you have this entire graph also as an edge list? Let's see what will happen if you, one thing that you can do is the simplest way to read this graph into something in memory will be to get a pair of strings. Every edge can be represented as a pair of strings. Each string is a gene name. Okay, so I can represent a pair, gene 1, gene 2, and I can put that into a vector or into an array, a dynamic array of edges. Okay, so the entire edge list could be read into memory as an edge list of size 1.3 million. Okay, 1.3 million, and I can have, then you say that, okay, I have my graph in my memory. Now, if you have that as a vector and as pairs of strings, now let me ask you simple graph questions. For example, is there a path between ZZZ3 and A1BG? Find the path on this graph by jumping from node to node. So it will be, for example, ZZ. Let me get a new slide here. Okay, ZZZ3 maybe somewhere. This APB. What, I don't know what the name was. I'm just going to write APB4, for example. Maybe they are connected to each other by a sequence of edges here. So there may be a path in between them. How do you find the path in between them? By running some kind of a graph algorithm, like the Dijkstra's shortest path algorithm. You want to run it on this graph. Or give me all the common neighbors of APB4 and ZZZ3. So, or whether, what is the average degree in this graph? Average degree, or find all the triangles in this graph, meaning that find all 3 pairs of nodes in which all 3 are connected to each other. So I can have different graph queries. But one thing I want to say is that if you just have this vector of, for example, how do you search for a gene in this vector of yours? You will have to say, okay, for example, since you have these pairs of genes, you either, if you have this vector, every time you want to know, for example, what is the degree of APOBEC 1, you have to go through your entire 1.3 million vector list, just inspect all of those pairs of strings that you have in your vector, and if one of them is APOBEC 1, increment its neighbor count by 1. Okay, so this query will take 1.3 million. It will take ON time. You will have to go through your, not ON actually, it's the number of edges time. So here, OE time, let's call it like this. So there are 2 things that's about complexity in a graph, which is you have the number of vertices and the number of edges. Okay, we know that the number of edges in our pathway commons data set is 1.3 million. But what about the number of vertices? We don't know that yet. Okay, let's try to find it out. Let's write a program to do that. I think I have a random walk program somewhere here. Let me try to find that out. I think in some teaching last year I did, not this one, so it's this year. Let me try to quickly go ahead and figure out where assignment 4, or maybe search for here. I should have, I don't want to write the program from scratch, or I could write that actually. Yeah, let's do that. Why don't we do that? Let's write the program from scratch. Here I will write the program, and or maybe I can still find it. My query was, why don't I just search in this teaching? Here I will find the code directly. Teaching random walk. So I had a program written in Java at some point in time. So let's see. Well, this is very old. I think there's a newer version of that. I have, but let's see. But what I want to tell is that this vector, asking for example, the degree of a node is going to take you OE time to give answer to that. What is the degree of a particular, what is the number of neighbors of a particular gene? And running Dijkstra, for example, on just edge lists like this, it's going to be very, very inefficient. Okay, don't want to go through your edge list all the time when you're doing things. The most efficient way, in my experience, that I've figured out to represent the graph is by using the adjacency matrix representation, actually the adjacency list representation. But let me show you how we can represent the same graph as a matrix. If each of these rows is a square matrix, for example, if each of these entries in my matrix are either zeros and ones, because in the pathway commons database there's the edge or not, it's an unweighted network. There are also, but in the string database, it was a weighted network. It was weighted with confidence. Now, actually, in pathway commons, the edges are directed. When we have a graph, we usually ask these things. A graph, given a graph, is it undirected or directed? An undirected graph or a directed graph. In directed graphs, the edges between two nodes, A and B, have a specific direction. So it's possible to go from A to B when you're finding a path, for example, but going from B to A is not possible on a directed graph. On undirected graphs, on the other hand, the edges are undirected like this. In the directed graphs, sometimes the edges are named as arcs. Okay, I'm writing it in the chat box. Arc is what these edges, directed edges, are called, but I just call them directed edges. And this is, in some networks, we may have undirected edges. And this direction, and whether they are directed or undirected, may be related to the semantics of the interaction. For example, if I tell you, if you look at this in, some of these data, SNRPA1 is incomplex with XAB2. Do you think this is a directed interaction or undirected interaction? The meaning of this verb, their relationship between them, tells us whether it's directed or not. For example, if it's incomplex with something else, it means that this one is also incomplex with this, right? So it's the semantics of this interaction. They're just basically saying that they're interacting. A and B is interacting together. So the semantics of this biological knowledge is undirected. So when I'm going to put an edge between SNRPA1 and XAB2, I'm going to put an undirected edge. But what about another one? Let's take a look at this one. DINK1LDYNC, so long or difficult G name to produce, controls state change of stack 2. So does it mean that stack 2 also controls the state change of this one? Probably not, right? I mean, this is the knowledge that is given to us here is directed. Therefore, when I'm going to put an edge between this dink1li1 and stack2, I'm going to put a directed edge here. So if you're going to model the entire pathway commons network, you're going to see that you will have to have both undirected and directed edges in your graph. But one way to represent these directed and undirected edges is what we call an adjacency matrix. And the other thing is whether the edges are weighted or unweighted. Okay, again, this is going to be whether it's weighted or unweighted is some property of your data set. For example, the pathway commons data set is unweighted. If you look at the edges, it just tells me the existence of an edge. There is no numerical value associated with the edge, and therefore the edges in pathway commons are unweighted. On the other hand, if you look at the string database, there is some numerical value associated with the edge, the confidence we have on the edge. Some edges we are really confident, some edges we are less confident. So therefore, instead of having just the edges zeros and ones, we may have confidence values written in this adjacency matrix. Okay, the adjacency matrix is, as you can see, it sizes whatever the number of vertices are, OV squared. So it will require this much memory in resources, okay, if you have a matrix like this. But we are going to see that storing the matrix with all of the entries like this that takes this amount of storage is really space inefficient. It will be computationally efficient, but it will be very space inefficient because the graphs that are given to us, for example, there are here 8.6 million, even this one, which is more dense than the string one, 1.3 million. Okay, are those sparse graphs or not? Sparse means that the number of edges, sparse means that a sparse matrix is a matrix in which many of the entries are zero. So if I have this adjacency matrix representation, how many of the entries? I'm going to have in the pathway commons example, in the pathway commons data set, I know that I'm going to have at least 1.3 million of these entries filled. Here's a question to you. Is it possible to, if I'm going to put the pathway commons information into this matrix, my edge list is, its size is 1.3 million. How many, can you tell me a range? How many cells in this matrix are going to be non-zero? Okay, so my number of edges, as you can see, is in pathway commons 1.3 million. Okay, with 1.3 million edges, are there exactly 1.3 million non-zero entries here, or something else? Do you have any answer to this question? How many cells on this matrix are going to be non-zero? How many of them are going to be one actually? Because the only value I'm going to put there other than zero will be ones, which shows the existence of an edge. Is it going to be exactly 1.3 million cells? Because I have 1.3 million edges. So what does a cell represent? Cell represents an edge between these two nodes, between these two vertices. So if there are 1.3 million edges, are there going to be 1.3 million cells exactly non-zero in this on this matrix? Any ideas? You may write yes or no, for example. Are there going to be exactly 1.3 million edges if my edge list is 1.3 million? What is your guess on that? No. Okay, no is the correct answer. What about, what is it then? Is it going to be larger than 1.3 million? Or less than 1.3 million? How many cells do you think? The answer the answer right now actually is not exact number. Okay, why is it not an exact number? Let me tell you this. If all of the edges that are here were undirected in nature, okay, in their semantics, if all of them were undirected, I would have exactly 2.6 million edges, because an edge in that table is going to be represented as one in two different cells. Think about it like this. Look at these proteins, protein I and protein J. If there's an edge between protein I and protein J, since this is a square matrix, there will be an entry related to protein J here and protein I here. This entry will also be 1. Okay? So each edge is going to, actually each cell here correspond to a directed edge from I to J. Okay? If I have undirected interactions, there will be exactly two cells that will be non-zero in this cell, so hence 2.6 million. But some of these edges are directed. Not all of them are directed, but some of them are directed. For directed edges, I will have each directed edge, since the other direction, for example, if there was a directed edge from I to J and not J to I, this entry here will be 0. Our matrix will not be symmetric. Therefore, the answer to how many edges here will be non-zero should be, the answer to that will be, it should be something between 1.3 million and 2.6 million. Okay, this is the upper bound. The lower bound is 1.3 million. If all of them were directed edges, I have 1.3. Oh, there's another question. It will actually, it may be less than 1.3 million if the same two genes, the same two interactors here, A and B, appear in more than one line, because there may be different interactions, right? For example, they may be in complex with each other, and they may also, for example, they catalyze successive reactions in a biological network. So we also don't know that. All right, maybe even less than 1.3 million. Not, we cannot say at least 1.3 million. But these are the things that you should consider when you're constructing your graph. For example, one thing you should definitely consider is that, are you going to also have these chemicals in your the compounds in your network too, or is it just going to be a network of biological network of genes? But let's try to have them as well in our database. In this last 7 minutes, I just want to write a very short program to read this file and put the, just want to know what is my vertex size, what is the size of my vertex, therefore whether this adjacency matrices will be possible to maintain in memory, because this is a really critical thing that I should consider, how much memory it's going to occupy. So let me look at this random walk. So yeah, I have a newer version of this random walk. Let me look at this one. All right. So I'm just going to pick parts of it. For example, let me just, so this is about random walk clustering. Let me copy this into a new file, and I'm going to erase many parts. I just want to, so this is a, I'm writing some Java program, but I will just need to, how I parse this pathway commons file. And I want to just show you that Java file, Java source file. Let's save it in desktop. Read network. So I'm just going to name it as readnetwork.java. Let me erase unnecessary parts. I will keep this. So the way we are going to use it is that read network, it will just get a graph file name and that's it. There's no output file. You're not doing anything else. You just read a graph, get a graph file name from the command line, and this is doing some sorting of things. I'm erasing this implementation of quick sort. I'm erasing that part too. Yeah, take the transpose of, so let's all erase these things. There's no update, normalize the matrix. I'm erasing this part too. This one too. So this is it's doing some clustering. Actually, I'm deleting many of these parts. Yeah, now. Oh, still quick sort, random walk. Yeah, I think I need to erase all these parts too. Anyway, sorry for taking, so we have only a few minutes left, but I will be able to read this one and give you the size of this vertex left. Okay. Wow. I think actually, yeah, it's just reading the graph file is just here. Okay. So we get the command line arguments. The length is, we want the length to be 1. We get our graph file name, and that's it. Okay, so we read our graph, we read it line by line, and we read it as long as there are lines. I think it's pathway comments is also tab separated. Let's see, I think it should be tab separated, yeah. There's a tab between 3 items. All right, so if it's, also I need to change the name of this to read network class. By the way, I'm writing in Java, but I just write it as a C code, not object oriented at all. I just use these. The reason I use Java is I could have written in C++ and use STL, but just being lazy, I do that. So my second protein actually is the second so when you split your line after you process them into tokens, if you split them by a tab, the first entry in this format is a protein or some actor name. The second one is the type of the interaction, and this one, the third one, which is index 0, 1, 2, the second index is your other protein. So we have this adjacency matrix that I'm maintaining, which is a hash table. Okay, so I don't need this, I don't need that. So I'm going to put these actors into my hash table. When I read them, if it's already in there, I don't do anything. I just put the new one as a neighbor to it. So this representation actually may be a little bit confusing. Let's just do it like this. I just put my, or maybe let's maintain it like this. I'm going to explain what this means later, but in order to read this file, all I need to do is read line by line. I read the first protein, the second protein, and I have my adjacency matrix, but the way I maintain this adjacency matrix is that actually is an adjacency list, which is a more efficient way than storing the entire matrix. Okay, and this one, we are done. We have read the entire table into entire graph into R, and the size of this hash table will tell me how many nodes there are in the network. So let me quickly compile it on my command prompt. Read network. Okay, so it compiled. Now we'll be able to see before Java read network, the pathway comments. So this was the file. So, okay, it's using, let me use Ubuntu for that. The Java runtime, and this one is in different places. So I go to users desktop. Now I'll be able to run it in here. Pathway code. Okay, now it's reading the whole 1.3 millions, and it did that in a couple of seconds, as you can see. And there are 30,000 nodes in the network. So we were able to find that the size of this V. Okay, so there are 30,000, and as you can see, it was very fast. If you don't use hash tables or a map to read this data and try to maintain this information, it may take minutes to read the network into the adjacency matrix. But 30,500. Now, what is this 30,000? If I'm going to maintain this as an adjacency matrix, it's going to be 30,000 times 30,000, 30K times 30K, it's 900 million. Okay, so 30K squared gives me, K squared gives me a million, this squared gives me 900 million. So there would be, if I was to maintain as this as an adjacency matrix, there would be 900 million entries in this square matrix, and only at most 2.6 million of them is non-zero. What does this mean? Your adjacency matrix is very sparse. Almost all of it is zero if you were to maintain it as a matrix like this. Okay, because there are at most 2.6 million non-zero entries. If you have a square matrix like this to represent your graph, you will just wasting up space for all those zeros. Okay, there are not the potential number of the maximum number of directed edges in such a graph is 900 million with 30,000 nodes, but we only have 2.2, so it's very sparse. So I'm going to show you a different way to represent this matrix as an adjacency list, and it's, as you can see, is very efficient. There are 30,000 nodes. Some of them are proteins, some of them are compounds. We are going to do some analysis techniques to, for example, maybe just report types of interactions, report only how many chemicals are there in this network, things like that. Okay, so I will stop here now, but the first thing, as you see, we have to consider is to find a way to represent this graph in memory, and this is how the way this implementation that I have here is an hash table of hash table or map of maps. The entire list of vertices is stored as a single hash table, and the neighbor list of a protein is also stored as a hash table, because if you want to ask whether is this protein a neighbor of this one, we want to also quickly look in that list of neighbors whether P3, for example, is a neighbor of P1. Okay, so this representation that I will discuss in more detail on Thursday is a graph representation which is called adjacency lists, and the lists are maintained as hash tables for greater efficiency of lookup of string names. All right. Oh, by the way, again, I hid myself behind these Ubuntu shells. Okay. All right. That's it from me for today. If you have any questions, I will stop recording now, but I will stay here to answer your questions if you have any. All right.